Part 2 of the October 29th Biology 105 lecture. We're back, everybody. Time to get a second phone, one just for recording. That would be sick. I could have different angles too. And yeah. Anyway, multicellular organisms. Cells divide for growth, cells divide for development. My camera angle's all messed up now. That's better. If you take some damage, all right, the cells need to repair. Cells renew themselves. So in general, like cells just get worse over time. They accrue gunk. The proteins undergo like intermediate levels of loss of structure as they age. It's good to have a new cell. New cells are more productive. New cells that carry out photosynthesis carry out higher rates of photosynthesis. They're more efficient. They have higher photosynthetic efficiency. New cells that carry out respiration have a higher efficiency of respiration. So that's the process of renewal. Take the mother cell, divide it, and you get two cells that are new. All right? So it's a process of renewal, process of repair to replace damaged cells or cells that have been removed, process of growth, process of development. Cells need to divide. Established? Any questions about cell division and why it needs to happen? Okay, because now we're going to start talking about how it happens. We're going to talk about mitosis and meiosis, and there is a nice old CG video available for you that runs through the steps of mitosis, but I'm not watching it again. I've watched it a dozen times. I'm not doing it. PowerPoint's available. You watch this video or 1,000 others like it that all run through the steps of mitosis, which I'm going to do with you in a lecture. And then if you want to watch the videos, find them. They are helpful, I swear. It is helpful to watch a video explaining mitosis, okay? But that's homework for you to do. They're 5 minutes long. Just check one out. It's going to be great. The cell cycle. All right. We've got two types of cell division. We have mitosis and we have meiosis. All right? Mitosis is sometimes referred to as replicative division, okay? Because when mitosis happens, you take one mother cell, and your goal when you carry out mitosis is to make two identical daughter cells, all right? So you want to go from one cell to two copies. These cells should be identical to one another, and the goal really is to have them be identical to the mother cell as well. All right? You're not trying to generate a bunch of variability. You've got something that works, and you're trying to make copies. Okay? So mitosis makes copies. Cool? Meiosis is the process for generating gametes. And this generates gametes for sexual reproduction. And the only reason for sexual reproduction to exist from an evolutionary standpoint is to generate variability. So in meiosis, variability is generated because you want there to be variability in the population following reproduction. So just remember, right? Like mitosis makes copies. All the processes in mitosis are designed to minimize variability. Whereas in meiosis, you're making gametes, which exist for sexual reproduction, which exists to generate variability. So in meiosis, the processes are geared towards generating more variability. Fair enough so far? Don't worry, we're going to hit it all in more detail too. Please, question.
Do you have any mnemonic devices to help memorize this?
Oh, gosh. You know, I really should, but I don't think I do. I just remember mitosis and meiosis, and I don't have any mnemonics. Does anybody know any good mnemonics for mitosis and meiosis? We got one.
Okay, I don't know if it's for this specifically, but I remember the actual cycles is PMAT. That's something.
Oh yeah, I mean, no, that is good. PMAT's great. I think because there's also cytokinesis. And so I think of a kid following like a nap, and they hold up a sopping wet mat and they go, PMAT, see? And we'll talk about that. It's a good mnemonic for remembering that. And if it's meiosis, you've got 2 of them, you know. But for like mitosis, making copies, and meiosis being reductive, I've never had a mnemonic, and I don't know a mnemonic, and it looks like polling the classroom right now, no mnemonic has ever existed for this memorization. But if you come up with a good one, please share it with the class next time, and you can get some John points for it. Awesome. Yeah. So we're going to be focusing this week on mitosis. That's the replicative one. Mitosis makes copies. Oh, you got one? Oh, same one as before? All right, fair enough. I saw a hand at the back. No? Okay. All right. Cool. Awesome. So we're going to be focusing a lot on mitosis to begin. Mitosis is the simpler one. And then we will talk about meiosis, which is kind of like two rounds of mitosis with extra stuff thrown on top. Yeah, question, please.
I was thinking for mitosis and meiosis, I don't know if this is like an actual thing, but when I think of it, mitosis has a T in it, and you could say two of the same.
Oh,
Mitosis,
yeah.
T means two of the same, and meiosis is just like phosphorylation, that means it makes the 4 non-identical.
Yeah, if you have a mnemonic for one of the 2 categories, then the other one is...
Eliminate the other one. So mitosis, two of the same. That's just what I'm thinking, but I don't know if that's helpful.
I'm going to give you 1.7 John points, and they round up to 2, because we only deal in full John points. So very good. Yeah, mitosis makes 2 cells that are identical. And meiosis makes 4 cells that are non-identical. I like that a lot. Mitosis, 2 cells. That's good. Now, what I will do is take that and use it as if it was my own idea and give you no credit. Is that okay?
Yeah, that's fine. I don't
Okay.
need credit.
I just thought of something great, class. All right, cool. Thank you. Mitosis, we're going to be talking about them. I'm thinking about toes. Because I always say, mitosis, go in my socksis. That's the joke I always make every semester. But now everything's all jumbled up. Okay, somatic versus germ cells. How does this relate to anything? Well, somatic cells are also called body cells. Soma means body. Y'all still read Brave New World? You're supposed to read that as a student, right? You read Brave New World? No?
No.
Some do, some don't.
I had to read it for AP, but
Oh, sick. Okay, cool. Yeah, you either have to read an AP, you got to read either 1984 or Brave New World, and then you get to have the debate of which one's a better view of a dystopian future. And then you read the letters between Huxley and Orwell, and then you do that, and that's the beginning of your intellectual blossoming. At least that was my lived experience, okay? So there's a drug they take in Brave New World, and it's meant to keep the lower ranked individuals comfortable and dumb them down and make them as if they were functioning parts of the body, but not really involved in anything, any big thinking or any reproduction or anything like that. I mean, they breed, but breeding's all done in machines anyway, so there's no, real sexual aspect. Check this out. Soma is the drug that they take in Brave New World. Mind blown, right? And that's the same root as somatic cell. Your somatic cells are your body cells. They're the majority of your cells. They are not involved in contributing genetic material to the next generation. They're not involved in shaping the way the population's going to work. What they do is they keep you alive, all right? They're in your liver, they're in your skin, they're in your muscles, they're your somatic cells. And what do? They routinely divide via mitosis. They make copies of themselves, all right? And they carry out most of the functions of your body. You've also got a special pool of cells called the germ cells. And these germ cells generate the gametes, okay? They generate the actual egg and sperm cells, the reproductive cells. And this terminology applies to more than just humans, but at this point in the course, we're going to go very human-centric because it makes for cleaner examples, I think. So I'm just going to talk about all of this from a human perspective. And if you got questions about how does this work in something that's not a human, just ask. It's totally fine. All right? Sick. So, somatic cells. Vast majority of the cells in your body right now. What do? They divide, they reproduce themselves. There are some pools of like stem cells that divide and make multiple cell types, but this is all division by mitosis. Not involved in generating gametes, and not really concerned with generating variability in the genetics. It's all about making copies. Okay? Somatic cells. Then we've got the germ cells, and the germ cells divide via meiosis. Now, I've found that some people have a familiarity with the concept of ploidy, and some people don't. This was from a previous lecture on transporting things from cell to cell, but it's a very strange shape, and I don't like, looking at it. I'm going to actually skip to the next page. All right. How's that look? Everybody can see that okay? Cool. So ploidy, let's talk about it. Human, all right? Standard issue human. Diploid organism, okay? Human generates a sperm cell. It is haploid. Or generates an egg cell. All right, I'm going to write egg in this so you can tell that it's an egg. All right, I'll also put like a belt on it, like this. And now it's like a Humpty Dumpty egg. All right. Very good. Now, both of these are examples of haploid cells. So in your body, you have 2 copies of every chromosome. 2 copies of each chromosome. All right. Which can sometimes be abbreviated as ohms, like that. Now, in the sperm cell, or the egg cell, and we'll put a little hat and a little face on the sperm cell too. All right, these have 1 copy of each chromosome. All right, 1 copy of each chromosome. So, when the sperm cell and the egg cell meet and come together, all right, they generate the zygote, which is now diploid. So your cells have 2 copies of each chromosome. Your germ cells have 2 copies of each chromosome. But the germ cells are going to divide via meiosis, and this is going to give you haploid gametes, which then can fuse to regenerate the diploid organism. Okay? So what does this mean? We've got all these different chromosomes, 23 distinct chromosomes. Oh, I meant to do this. 23 distinct chromosomes, all right, that exist in your body as pairs. So you have 2 copies of chromosome 1. You have 2 copies of chromosome 2. You have 2 copies of chromosome 16. All right? The 23rd set of chromosomes are the sex chromosomes, and you can have an X, you can have an X, an X, an X, a Y. There are people who have XXY and XXX and YY. Well, not YY is not usually viable, but XYY. There's all these different genotypes for the sex chromosomes. There's variability here. But as far as chromosomes 1 through 22, you've usually got 2 copies of each, except for in cases of trisomy, which we will talk about later. But this is the karyotype of a diploid organism. 2 copies of each chromosome. Yes?
Are they numbered in their order for any specific reason?
Size.
Okay.
Yep, it's all based on size. So the largest chromosome is chromosome 1, and the smallest one is chromosome 22. I mean, the Y chromosome's technically smaller than chromosome 22, but the sex chromosome pair is distinct. Anyways, yeah. Cool. So this is what we call a karyotype. And a karyotype is an actual picture, okay, of all of the chromosomes in a regular diploid cell. All right, there's a human cell, all the chromosomes laid out. All right, 2 copies of each. Now, what do we know from this karyotype here? We can't be sure that it's a human, okay? We can't be sure of that. All we can know from this karyotype is that the organism has 23 chromosomes, all right? And that it's got diploid cells, that this cell is diploid, all right? That's kind of like a tricky aspect of this, is there are other organisms that have different numbers of chromosomes. Just because you got 23 chromosomes doesn't necessarily make you a human in the world out there. All righty. Yes, please.
Are they normally colored like that, or is that just differential staining?
Great question. So it's not even stained, it's post-production, right? So they colorize this image. So you can do fluorescent staining to identify the chromosomes, but then you usually actually just take the picture of all the chromosomes laid out and then manipulate their location in like Photoshop and then slap colors on them to differentiate between them. Yeah. So this is just a colorized image. All right. So prokaryotes and eukaryotes. Let's talk about their genetic material. If we're talking about cell cycles, we have to talk, let me double back really briefly right here. If we're talking about cell cycles, we're going to be focusing on tracking where the genetic material goes throughout the cell cycle. So this is an actual colorized image of genetic material during mitosis. You start with a big pool of genetic material, the chromosomes sort of condense up, and then they get split apart, and then they decondense, and that's mitosis, all right? I'm gonna get into a lot of detail about it, but I just wanna let you know that we're gonna be focusing on the movement of the genetic material, because that's really the main focus of the cell as well. Okay, prokaryotes versus eukaryotes, what do their genomes look like? What is a genome? A genome is all the DNA in a cell or in an organism, okay? Depends on the scale that you're talking about. Remember when we talked about metabolism? And we could talk about the metabolism of a cell or the metabolism of an organism. But we could talk about an organism's genome or we could talk about a cell's genome. Usually, if you're talking about an organism's genome, you look at a single cell and you look at that genome, right? Anyway, because most of your cells should be identical copies of one another, at least for the genetic material, because they were all generated via mitosis. There can be regulation of the expression of that genetic material that differs between cell types, but the genetic material available, the actual genome, should be the same from cell to cell to cell to cell to cell in all of your somatic cells because they were all produced via mitosis. All right, prokaryotic genome versus a eukaryotic genome. Prokaryotes are simpler, right? They don't have to have as many instructions programmed or as much regulation of their instruction kit because it's one cell and all the genes they've got, are going to be expressed or not expressed. They don't have as much intervening genetic material that's involved in regulating gene expression. Think about the difference between a multicellular organism and a unicellular organism, right? You've got a lot of different cell types, and if the same blueprint is present in each one of those cells, there has to be a lot more going on in regulating which regions are getting expressed, all right? So one of the things that allows for all that regulation is a more complex genome with a lot of DNA set aside just for regulation. You're a single-celled prokaryote, no worries. You don't have to worry about all of that as much. There's still regulation of gene expression, but it's not quite as complicated as in a multicellular organism where you have to develop wholly different cell types. So why do I say any of that? Why do I do any of that stuff? Because we're going to talk about the density of genes on the chromosome and all that. And I want you to see why a simpler genome, a physical structure for the genome, works for a prokaryote. So in prokaryotes, the entire genome is a single chromosome. It is a single circular chromosome that gets all jumbled up, all right, in the center of the cell. They don't even have a nucleus. They don't even separate it from the dang cytoplasm, all right? Whereas in a eukaryote, you've got a bunch of discrete chromosomes, and each one is a big, long strand of DNA that has a complicated structure where it gets condensed around these histone proteins and gets all packaged up, all right? So prokaryote, the entire genome's one chromosome, and it's circular. Eukaryote, a bunch of linear chromosomes. I threw a lot of stuff at you there, and I definitely felt myself starting to ramble. I checked out for a second. Did that make sense? Can I clarify anything that I've spoken about so far? Okay, then we'll keep on moving. We'll keep on crushing it. So, this is where the terminology is going to start getting really annoying. Chromosomes, chromatids, centromere, centrosome, chromatid. These are all different things, okay? And you have to be able to identify the differences. You have to be able to separate all those terms. I'm going to work really hard to keep my terminology consistent as we move forward so that it all makes sense, okay? So check this out, right? Your genetic material, because we're, like I said before, we're going to be taking a human, an anthrocentric view here, okay? We're going to be looking at diploid eukaryotes, okay? So as a eukaryote, your DNA is often in this dispersed form where the long strand is just spread out. All the different chromosomes are sitting together in the nucleus, all spread out like a big bowl of spaghetti, okay? But sometimes when it's time to equally divide up that big bowl of spaghetti, all right, you take each noodle and you roll it into a little ball, all right? That way you can just easily separate, like you each get one of these, and you get one of these, and you get one of these, which is easier than taking the entire blob of spaghetti and pulling it apart and being like, okay, wait, here's noodle A, and where's noodle A over here? I got to get one noodle A there and one noodle A there, all right? Instead, you ball it all up to make separating easier. So, if I'm going to take one cell and make two identical daughter cells, what do I need to do? I need to take all the DNA that was in that mother cell and duplicate it, okay? I got to make copies of each chromosome. So does that make sense? Cool. Because I start with a cell that has two copies of chromosome 1, and I'm doing mitosis, so each daughter cell needs two copies of chromosome 1. So I've got to duplicate the chromosomes. That's one thing that must happen. Also, in order to make separation of the chromosomes possible in an organized way, the long spaghetti strands need to be condensed. So the chromosomes need to be replicated before division, and also the chromosomes need to be condensed before division. Do those two requirements sort of make sense for everybody? Wonderful. What we're looking at right here is a replicated chromosome. So if you look at the karyotype that we saw before, these images, even though chromosome 2 especially looks like a replicated chromosome joined together, it's not. It's two separate chromosomes, two separate copies of chromosome 2. Here's a replicated chromosome, all right? So this was chromosome, let's just say it was chromosome 1, all right? Imagine half of this was chromosome 1. And then, during the cell cycle, all right, that whole chromosome got replicated. So now you had two copies of chromosome 1. But they're both stuck together, okay? And then they condense, and they make this replicated chromosome structure that now has two sister chromatids joined at a centromere. So here's what it's like, all right? My finger now is chromosome 1. It's time, I'm getting ready for division. So I need to make 2 copies of chromosome 1. So I take chromosome 1 and machinery in the cell, all right, makes a copy of it. And now there's 2 copies of chromosome 1, and they're sort of stuck together. And they're joined at this centromere, okay, this little protein structure that holds those 2 chromosomes together. Because later on, when I make the daughter cells, I want to make sure each daughter cell gets a copy. So I don't want these to separate and get all mixed in with the bowl of spaghetti. I want them to stay connected so that later I can easily say, I can pull this out of the bowl and go, oh, cool, that's chromosome 1. Let me just rip that apart and give 1 to each daughter cell. It's very complicated. Yes?
So basically, one chromosome, there has to be 2 of them because one of each makes one daughter cell.
Yes.
So you have 2 chromosomes that are the same, and then each one makes a daughter cell, so then you have 2 daughter cells.
Exactly. So this is really big, right? And this is where it gets super complicated. Because now, I mean, I've laid that out for you, and it's like, okay, that might work. But now, remember that you're diploid. So you've got chromosome 1A that does this, and then chromosome 1B that also does this. So now you've got 2 replicated chromosome 1s in the mother cell. All right?
Chromosomes in the mother cell.
Yes, but a replicated chromosome, this thing, is still called a chromosome. So we didn't increase the ploidy number at all. It's very annoying. Bear with me, folks. Chromosome 1A undergoes replication, makes a replicated chromosome. All right? It's a replicated chromosome joined at the centromere. It is still 1 chromosome. This is still chromosome 1A. Chromosome 1B undergoes replication. All right? Boom. Makes a replicated chromosome joined at a centromere. This is still chromosome 1B. So now the mother cell went from having chromosome 1A, chromosome 1B, to having chromosome 1A and chromosome 1B. It is still a diploid cell, and we talk about these replicated chromosomes as each being a single chromosome, made up of a sister chromatid and a sister chromatid joined at a centromere. And each sister chromatid should be identical to the other sister chromatid. Yes?
Wait, so I guess I got more confused.
Totally.
Are there then 4 sister chromatids per 1 chromosome? Because you're saying 1A makes 1A, and then 1B makes 1B, and then
So there are now 4 total sister chromatids for chromosome 1.
so there's 4 sister chromatids and 1 chromosome.
There's 2 chromosomes.
2 chromosomes, but so it's like, okay.
Chromosome 1A and chromosome 1B are similar to one another, but one came from the sperm and one came from the egg, so they're not identical.
Okay.
That's the difference.
Okay, so then you have 2 chromosomes.
2 chromosomes still. It's 2 chromosomes to 2 chromosomes, but now these are replicated chromosomes that have 2 sister chromatids each.
And they're diploid because they have the 2 chromosomes.
They were diploid like this, and they're still diploid like this. Maybe the next slides, I hope they'll help. This is all, we're really like, listen, we got 49 slides total to cover in like 3 lectures. So I'm intending this to take, I'm trying to go slow. We'll take our time, and I'm going to explain it in many different ways, and one of these ways will hopefully work for you. So if an explanation feels like it's just not finding purchase in your mind, then, try to get through it, and we'll find another type of explanation that does. All right, DNA and chromosome structure. So you got the DNA double helix that we've kind of spoken about already when we talked about biological macromolecules. Here are some pictures of replicated chromosomes. How are the 2 related? All right, how do we get from this long double helix strand to these replicated chromosome blob things? Okay, we're going to talk about that right here. the DNA double helix, all right? It's just the polynucleotide, right? It's a series of nucleotides all joined together via dehydration synthesis into a polymer. You have base pair complementarity, right, where A binds to T and C binds to G, things that we've spoken about already, but I promise you we will review thoroughly when we hit our genetics portion of the course in earnest. All right, it's gonna be great. You're gonna love it. All right, so you've got the long polynucleotide DNA double helical structure. This molecule gets wrapped around these proteins called histones, all right, and that helps to condense the DNA up a little bit, okay? Then that DNA, which has been wrapped around histones, all right, gets further condensed and gets wrapped around these additional proteins, all right? So it's kind of like, back in the day when I started teaching this, I had wired headphones, you know, and I would demonstrate it by pulling the headphones out of my pocket, and I would say, like, look at these headphones. They're all tangled. They're annoying. I hate them. And then I would wrap them around each of my fingers, okay? And that would say, oh, look, I have a nice packaged system. Oh, perfect, exactly, yeah. Beautiful, thank you so much. Look at all this spaghetti, all right? So I've got the long thing, this is great. I got the linear DNA molecule. If I had 46 of these in a box, that'd be a nightmare to make sure that the two people each got 23 of them, and that each person you got a red one, you got a red one, you got a blue one, you got a blue one. So having the DNA all stretched out like this is useful when you're trying to read the messages on the DNA, right? But it's a challenge when you're trying to move the chromosomes around and make sure that daughter cells get the appropriate chromosomes. So the DNA gets wrapped around histone proteins, like so. All right? And that makes it way easier to package and move. And then, you know, imagine I had 20 fingers and I did this, and then that gets further condensed and further packaged. And that's how you go from a long molecule of DNA to these condensed chromosome structures that we can actually visualize. All right? So you've got this long DNA molecule, and I don't want to throw these because I'm afraid I might hit somebody in the eye, but I'll put them right here for you. Yeah, awesome. Thank you for the sharing. You've got the long chromosomes, all right? They're all like long strands of string, and you wrap them around these proteins to help to condense them, right? Now, the long strand, okay, is called chromatin, all right? The chromatin fibers. Cool? So chromatin is different from chromosome, is different from sister chromatid. Isn't that great? Chromatid, chromatin, chromosome, blah. It all gets very frustrating. Now you gotta know chromatid and you gotta know chromosome, but chromatin, I don't think I'm gonna probably really test you on, okay? Good. So I'm hoping this helps. Check this out. We've got a single chromosome here. This is chromosome 1. This is chromosome 1A, all right? It's all condensed up, okay? So you can look at it. There's going to be a stage of chromosome duplication and then recondensation. It's a whole thing, okay? But here's now a duplicated chromosome or a replicated chromosome, okay? Here's a replicated chromosome. Now, this is chromosome 1A, and this is chromosome 1A, but right now, because they're joined together at a centromere, this is a sister chromatid, and this is a sister chromatid, or this is a sister chromatid, and this is a sister chromatid right here. The entire structure is still chromosome 1A. It's just now the replicated form of chromosome 1A. So this is chromosome 1A, this is chromosome 1A, and then before division or during division, the sister chromatids are going to get yanked apart, and then this is chromosome 1A, and this is chromosome 1A. Yes, please.
So
Exactly, that's exactly right. Yep. So we go, this is a chromosome. There's no sister chromatids involved at all. It's just a chromosome. Duplication happens to get the replicated chromosome, and now you have a single chromosome with 2 sister chromatids. Then those sister chromatids separate, and now you have 2 chromosomes, all right? Which, if everything went according to plan, should be identical to the initial chromosome. Because this is all mitosis, and in mitosis, we're only concerned about making, just think T for 2 identical daughter cells. See, I stole it already, but I thought of that. Yeah, so easy. It's great. Stealing ideas, it's a tradition in science. It's really fun. All right. Let's talk about homologous chromosomes. Let's do it briefly right now, and then we'll start here on Friday. Okay? So you know how I've been doing like chromosome 1A, chromosome 1B, all right? One of those chromosomes came from the sperm, one of those chromosomes came from the egg, right? They're both chromosome 1, so they both should have the same basic instructions on them, but because they originated from different people, there's going to be slight differences there. Maybe chromosome 1 contains the gene for eye color. It's not that simple, but bear with me. And you received a green eye gene from the sperm cell on chromosome 1, and you received a brown-eyed gene from the egg cell on that chromosome 1. And then they came together, and now you have 1 copy of the gene for green eyes and 1 copy of the gene for brown eyes. And they're both still on chromosome 1. That indicates that these chromosome 1s are slightly different, okay? We call that pair of chromosomes homologous chromosomes, all right? Homologous chromosomes is where we are going to start on Friday. Thank you all for your continued attendance, participation, positivity, and just overall, overall making my day brighter. Have a great day. Have a great Wednesday. Let's get through week 10.